After a deep study of the microbiome, I've come to the realization that there are two key things we have to do to recover a healthy microbiome that almost no one is talking about. We have to recover the lactic acid bacteria in the proximal colon, and we have to lower the pH of the distal colon to below 6.6. These two issues are the difference between a metabolically protective and health-promoting microbiome and a colon full of toxic acids, poisonous gases, putrefaction, and inflammatory bacteria. So let us begin at the beginning. As science began to understand the importance of the microbiome to health, focus was initially placed on probiotic supplementation. It was thought that antibiotic overuse was causing the loss of key bacteria from our microbiome, and all that was needed was to repopulate the microbiome with good bacteria. With advances in genetic testing, we learned that while things like antibiotics, chlorinated water, and glyphosate were indeed very destructive to the microbiome, it was rare that any particular good bacteria would be completely wiped out. Rather, the good bacteria would be decreased in number, and those that remained would be in a dormant state. It was then reasoned that if the needed bacteria were still there, then perhaps what was needed was to feed them with prebiotics. This way we could wake them up from their dormant state, and they could repopulate the colon. Prebiotics, such as those found in our Panaceum product, have proven very helpful, but again can only take us so far. It turns out that when good bacteria decrease in number and activity, bad bacteria take their place, and once the bad bacteria get a foothold in the digestive tract and establish a biofilm, it's nearly impossible to get rid of them. So I prayed, going through the medical journals and studies looking for a way to knock down the bad bacteria, but everything I found that killed bad bacteria killed even more good bacteria than bad. Then one day I found the answer in a single sentence in a medical journal. It would have been really easy to miss that one line in all the technical jargon of the study, but there it was, the key to dealing with bad bacteria and recovering the microbiome. The protocol is really simple. But to understand why we need the protocol as I've designed it, there is some biology we need to go through first. First, let's talk a bit about the nature of the bad bacteria we have to deal with. Bacteria can be roughly divided into two types based on the makeup of their cell walls. We have gram-positive and gram-negative. As a general rule, gram-positive are good for us and gram-negative cause us trouble. Let's take a trip down the digestive tract and see if this holds true. In the mouth, it is predominantly the gram-negative bacteria that cause gum disease and tooth loss. In the stomach, it is gram-negative bacteria H. pylori that cause stomach ulcers. In the small and large intestines, it's the gram-negative bacteria of the Enterobacter genus that cause disease. And it's not just the digestive tract either. Gram-negative E. coli cause most prostate infections and UTIs. Gram-negative salmonella can cause gallstones. Gram-negative proteobacteria can cause arthritis, and so on. Now, there are some good gram-negative bacteria, but even these we want to keep to a minimum. Gram-negative should be no more than 10% of the microbiome, in my opinion. Unfortunately, in many of us, it's more than 50% of our microbiome. The difference between these bacteria lie in what their membranes are made out of. The gram-positive bacteria cell wall is made up of a protein sugar mix called peptidoglycan. The gram-negative bacteria cell wall is made up of a fat and sugar mix called lipopolysaccharide, or LPS for short. The gram-negative bacterial membrane is much more complex than the membrane of the gram-positive bacteria, and it's this complex membrane that makes gram-negative bacteria so resistant to both natural antimicrobials as well as antibiotics. This complex LPS membrane is why almost anything we do to knock out the bad gram-negative bacteria ends up causing even more harm to the good gram-positive bacteria. Okay, well, what makes gram-negative bacteria so bad for us? Gram-negative bacteria can generate inflammatory lipids, toxic acids, poisonous gases, and putrefying amines. All right, let's look at these toxin classes one by one. First, the inflammatory lipids. The same LPS membrane that makes the gram-negative bacteria so difficult to kill is also one of the most inflammatory substances known to man. When the immune system sees high levels of LPS, it thinks that it is dealing with a runaway infection, and so it goes into an emergency state, battle stations. The way the immune system kills bacteria is by inflammation and free radicals, and this causes collateral damage to us. But it's better than a runaway infection that can kill us. It's a good system, and when a true infection has run its course, inflammation stops and the body repairs the collateral damage. In the case of chronic LPS exposure, the immune system continually keeps us in a state of inflammation and free radical damage. So, LPS from gram-negative bacteria is highly inflammatory and dysregulates the immune system. LPS also causes cell membranes of the body to leak. You probably already know about leaky gut, but that's not the only organ that can leak. LPS can cause the blood-brain barrier to leak. It can cause the blood-spinal cord barrier to leak. A leaky pericardium, which protects the heart, can be caused by LPS. It can cause a leaky peritoneum, which protects the liver. It can cause leaky capillaries, which causes a drop in albumin. And with a loss of albumin, in addition to edema, we can have drops in IQ, a decreased lifespan, a decrease in physical beauty, and an increase in cancer. And LPS for women causes a leaky mesoovarium, which is the membrane that protects the ovaries and the eggs. While leaking membranes in the organs causes trouble over time, leaky blood vessels is hemorrhage and is a medical emergency. So to avoid this, the body seals membrane leaks with a kind of glue. It's called an LDL lipoprotein A. Lipoprotein A is the sticky bad cholesterol associated with heart disease. If you have high LDL, the question is why, and the answer may well be your body is responding to membrane leaks caused by the LPS from the bad gram-negative bacteria. What else does LPS do? It damages mitochondria. It raises blood pressure. It causes premature cellular senescence. In other words, it makes us age faster. The next set of toxins created by bad bacteria are toxic short-chain fatty acids. I classify short-chain fatty acids, or SCFAs, into three categories: good, intermediary, and toxic. The good short-chain fatty acids are acetic acid, propionic acid, and butyric acid, and they are created by the good bacteria. These are all very similar, except they have 2, 3, and 4 carbons, respectively, so we have the same thing, just a small, medium, and large version. The intermediary short-chain fatty acids are lactic and succinic acids. In a healthy microbiome, they are eaten by other bacteria, which then in turn change them into good short-chain fatty acids. They only become toxic if there is a lack of the good bacteria in the gut to eat them, and they accumulate. Then there are the toxic short-chain fatty acids like formic acid, valeric acid, and isovaleric acid. Toxic short-chain fatty acids are inflammatory, imbalance the immune system, cause diabetes, create ammonia, and lead to brain fog and Parkinson's. They also cause heart, liver, and brain damage. These toxic short-chain fatty acids can reverse the Krebs cycle and cause cancer. In a normal Krebs cycle, raw materials are burned to create the energy we use. When the Krebs cycle goes backwards, energy comes out of us and is used to create raw materials that cancer uses to grow. All right, the next set of toxins I want to talk about are the putrefying amines. One such amine, TMAO, decreases our ability to clear cholesterol by 30% and increases the foam cells that make up plaque in the arteries, which is likely the reason it is associated with strokes, atherosclerosis, and heart attacks. TMAO can raise the colon pH 1,000-fold, which you will see later is a huge problem. The amines putrescine and cadaverine, in addition to being toxic, are responsible for the terrible smell of decaying corpses. An interesting side note, these cadaver smells increase threat behavior in people. In other words, if people smell cadaverine and putrescine on your body and breath, even at a subconscious level, they may treat you or see you as a threat. This means they may become angry at or afraid of you without knowing why. Finally, we have ethylphenol, or 4-EP, which is known to demyelinate the nerves and the brain and is associated with autism and other neurologic conditions. What do all of these have in common? They're made from animal proteins that weren't absorbed by the small intestine and were putrefied in the large intestine. Cadaverine comes from lysine, putrescine comes from ornithine, proline, and arginine, TMAO comes from choline and carnitine, and ethylphenol is made from tyrosine. It's not that eating meat is bad. It's eating more meat than you can digest and absorb and having a toxic colon. It's the spillover into a toxic colon that causes the trouble. As a side note, since excess amines are detoxified by the same systems that clear out histamine, if you have a high histamine level, it may be partially due to high levels of putrefactory bacteria like Enterobacter and Clostridia in your gut. Okay, the last set of toxins I want to address are the poisonous gases methane, hydrogen sulfide, and ammonia. In a healthy colon, good bacteria make carbon dioxide and hydrogen as intermediary gases, which are then turned into the good short-chain fatty acid acetic acid by actinogenic bacteria like Clostridia. However, if we don't have enough Clostridia, or the healthy type, to do this, hydrogen and CO2 gases build up. Some gets breathed out, some passes out of the body as flatulence, and some feed the methanogens, which turn CO2 and hydrogen into methane. In the gut, methane paralyzes the intestines and can cause constipation and/or cramps. When it gets into the general circulation, it can cause memory loss, headaches, fatigue, eye problems, abnormal emotions, rapid breathing, rapid heart rate, fainting, and premature cellular senescence, that is accelerated aging, just like LPS does. Another poisonous gas is hydrogen sulfide, also dimethyl sulfide and mercaptan, which is made by sulfate-reducing bacteria combining sulfur in the diet with hydrogen. Hydrogen sulfide and that family of gases cause sleep apnea, headache, irritability, insomnia, fatigue, stomach upset, DNA damage, and is responsible for the rotten egg smell of flatulence. The last poisonous gas I want to discuss is ammonia. Ammonia is formed by the deamination, which is the removal of ammonia, from an amino acid or protein. In a healthy digestive system, protein is digested into amino acids and absorbed in the small intestine. But if a person eats more protein than they can digest, it spills over into the colon, especially in the absence of a bacteria's preferred food source, namely prebiotics, and the result can be ammonia production. Good gut bacteria have enzymes to detoxify ammonia, but they can only do so much. When they reach their limit, ammonia can go to the liver. The liver then tries to lower ammonia using many of the same enzymes that the good bacteria uses, ALT, AST, GS, and GDH. But over time, the liver becomes damaged, and eventually ammonia spills out into the general circulation. It goes into the lysosomes and damages the body's cellular detoxification systems. Lysosomes operate at a pH of 4.5. If you raise the pH to just 5.2, it can't break down toxic fats. That means lipase is dropped by 60%. And if you raise it to 5.5, it can't break down toxic proteins. Catechin D proteases drop 80% at this point. Ammonia is also specifically damaging to the brain and can cause memory loss, ADD, insomnia, Parkinson's, Lou Gehrig's, MS, and other neurologic problems. Like methane and LPS, ammonia also causes premature senescence. Ammonia also directly feeds cancer cells. Finally, and perhaps most importantly, ammonia, which is an alkaline substance, causes the microbiome to go alkaline, and this, it turns out, is the key to the whole problem. Earlier in the video, I mentioned that I discovered a simple secret to knocking out the gram-negative bacteria and supporting good gram-positive bacteria and recovering your microbiome. Now I'm going to tell you the secret. It turns out that if you can get the microbiome pH to go to 6.6 or lower, there is a weakness that gram-negative bacteria have that we can take advantage of. At 6.6 and lower, good short-chain fatty acids can get pumped through the otherwise impenetrable LPS membrane of toxic gram-negative bacteria. Since the good short-chain fatty acids are only mild acids, they don't trigger the bad bacteria's heat shock protein defense mechanisms. Over time, the pH of the toxic bacteria get more and more acidic until they either die or go dormant. The gram-positive bacteria, on the other hand, aren't bothered by the good short-chain fatty acids. They can pump them out or neutralize them with potassium. Because of this, the good bacteria in our microbiome does everything it can to keep the microbiome from going above 6.6. That's the line in the sand, the Rubicon, for the microbiome. Below 6.6, ammonia, which is NH3, is converted into ammonium, which is NH4, which is a weak acid. Furthermore, ammonium isn't well absorbed like ammonia, so it won't go out of the colon into the liver and general circulation. However, if the pH goes above 6.6, ammonium does turn into ammonia, which is alkaline, and all heck breaks loose. Once the microbiome pH goes above 6.6, this alkalinity makes toxic gram-negative bacteria virtually impossible to get rid of, and they continue to churn out inflammatory lipids, toxic short-chain fatty acids, putrefying amines, and the poisonous gases that are causing us so much trouble. So that's the first key. That's the sentence I found in the medical journal that was the secret to recovering a good microbiome. You can knock out the bad gram-negative bacteria and all the problems they cause if you have a colonic pH below 6.6 and good short-chain fatty acids at the same time. But before I tell you how to do that, let's see how all this plays out in the colon. In a healthy ascending colon, we have gram-positive Firmicutes and gram-positive Bifidus, both of which make lactic acid. This lactic acid takes the alkaline stool from the small intestine and lowers the pH down to about 5.5, which suppresses methanogens and favors acetic acid production. At the same time, Bifidus directly competes with any fungi that would otherwise grow here. It can do this because Bifidus grows 2.5 times faster than fungi, and the fungi prefer a neutral pH. We actually have a very intimate relationship with some strains of Bifidus, one in particular called B. infantis. It's the only bacteria that has the genetics to consume all of the prebiotics found in human breast milk. It is the first symbiont that forms what becomes our microbiome. As we move into the transverse colon, the lactic acid from the lactic acid bacteria feed Firmicutes such as Clostridium, which make the majority of our butyric acid. As the lactic acid is converted into butyric and acetic acid, the pH rises to about 6.0. This is because butyric and acetic acids are less acidic than lactic acid. As we get to the descending colon, Firmicutes transition into the gram-negative Bacteroides and Akkermansia, a good version of chlamydia. This is where propionic acid is mostly produced. With the last of the lactic acid converted into propionic acid, another weak acid, the pH rises to the low 6s, 6.2, 6.3. At least, that's what's supposed to happen. In a pathological microbiome, it goes down a different path. Antibiotics, pesticides, chlorinated water, and a lack of breastfeeding mean lower Bifidus and other lactic acid bacteria in the ascending colon. Without sufficient lactic acid bacteria, Candida, fungi, can colonize the area. In the absence of prebiotics, the Bifidus that is there can make formic acid instead of lactic acid. Formic acid can cause the Krebs cycle to go in reverse, which is how cancer starts. Without the lactic acid, the pH won't be as acidic. That, combined with the formic acid, can cause the growth of methanogens. Thus, methane is produced. This has a paralytic effect on the intestines and can slow transit time 2.5 times, which further increases the alkalinity. The pH can rise to 6.0 or higher. As we move into the transverse colon, insufficient lactic acid lowers the numbers of Firmicutes, which make up most of our butyric acid. Thus, butyric acid levels drop. The gram-positive Firmicutes are replaced with the gram-negative Bacteroides too soon, and the pH rises to 6.6 or higher. The lack of short-chain fatty acids and the rise in pH can also trigger good bacteria to go rogue. An example is good Clostridia shifting into pathological C. difficile. Increasing gram-negative Bacteroides also increase our lipopolysaccharide, or LPS levels, and all the problems associated with LPS-induced inflammation. As we move into the descending colon, the pH goes above 6.6 to 7, 7.5, even 8. Acidic ammonium turns into alkali ammonia. Gram-negative Bacteroides growth is further stimulated by the ammonia, and they take the ammonia and carbon dioxide and create toxic succinate and hydrogen. This lowering of the CO2 and raising of the hydrogen triggers even more growth of methanogens. The methanogens eat our acetic acid, so we have less in our bodies. This increasing toxic methane level and sulfur-reducing bacteria increase toxic hydrogen sulfide levels as well. The ammonia that escapes the microbiome then damages the liver, lysosomes, and brain. The microbiome is now filled with toxic gram-negative bacteria, inflammatory lipopolysaccharides, poisonous gases, and the high pH makes it virtually impossible to do anything about it. Okay, that's the bad news. Here's the good news. We can return our microbiome to a healthy state. And here's what we have to do. 1. Clean bad bacteria from the mouth, because otherwise we're continually swallowing bad bacteria and reseeding our gut with bad bacteria. 2. Restart lactic acid production in the ascending colon. 3. Reacidify the colon. 4. Increase short-chain fatty acids. 5. Shift the gram-positive. 6. Get rid of the toxic fatty acids and poisonous gases. 7. Get rid of the toxic amines. And 8. Reclaim the new territory for good bacteria. Let's start at the top, the mouth. Using alcohol-based mouthwashes is a bad idea. Alcohol kills more gram-positive than gram-negative, and once it starts evaporating in small amounts, alcohol actually feeds gram-negative bacteria. If you have access to a medical ozone machine, you can rinse your mouth out with ozonated water every morning and evening. Ozone is the only disinfectant I know of that kills more gram-negative than gram-positive. If you don't have access to an ozone machine, you can take a capsule of our ellagic acid product and mix it with a little bit of water and swish it around your mouth before you go to sleep each night. Polyphenols, like ellagitannin and gallotannin, not only kill gram-negative bacteria, but also feed good gram-positive bacteria, so this is a win-win. Second, we need to restart lactic acid production in the proximal colon. I mentioned that there are 2 keys to recovering the microbiome. This is the second key. There's a group of bacteria called lactic acid bacteria. You would know them as Acidophilus and Bifidus. The lactic acid bacteria are responsible for making the lactic acid, which lowers the pH of the colon to as much as 5.0. This makes it difficult for candida and bad bacteria to grow. Then as the lactic acid is replaced by the acetic, butyric, and propionic acids, the pH will gradually go up to 6.0, 6.2, 6.3 before reaching the end of the colon. It's this initial dosing of lactic acid that sets the stage for the rest of the colon. How can we support the lactic acid bacteria? Well, mother's milk contains prebiotics that only Bifidus can eat. Then we are weaned, and our intestines are supposed to secrete prebiotics for these bacteria. But 20% of us don't have the FUT2 gene responsible for the intestinal secretion of the lactic acid bacteria prebiotics, with an additional 25% of us having only limited capacity for this. So some of us will have to do it through diet or supplementation. However, if the bad bacteria have a foothold in the distal colon, the ammonia they make can override the acids made by the lactic acid bacteria. Next, we have to reacidify the distal colon. We also have to increase the good short-chain fatty acids. If we can do this, we can shift the microbiome to a gram-positive dominant state, which will get rid of the toxic fatty acids and the poisonous gases. So all 4 of these can be accomplished with a single protocol, and that is a short-chain fatty acid microenema. As little as 1 ounce of acetic, butyric, and propionic acids in the right ratio and at the right concentration can both reacidify the colon and deliver the good short-chain fatty acids. And that takes us back to that 1 key sentence. Good short-chain fatty acids can kill or suppress bad gram-negative bacteria at a mildly acidic pH of just 6.6. The toxic amines can be addressed in the same manner, since once your gram-negative bacteria start decreasing and your pH goes down, you'll make less of these toxic amines. Once the bad bacteria have been killed or are dormant, all you need to do is feed the good bacteria with a prebiotic like Panaceum, and your once dormant good bacteria can now reclaim the territory it has lost to the bad bacteria and recolonize your microbiome. Okay, what does the protocol look like? Before you start, check your stool pH. If you can do a $500 stool test, great. If not, all you need is some pH paper. You want to get pH paper that has a range of 6 to 8. You'll press it against some stool and get a reading. I like to see a pH of 6.0 to 6.3. It's difficult to know what perfect stool pH is. I haven't seen any data on the pH of the stool of primitive hunter-gatherer tribes. We know that at 6.6, we begin to see ammonia and methane production, and bad bacteria start to grow. So I would say 6.6 is as high as you want and probably too high. So first, take the 6 to 8 pH paper and press it against some stool. If the stool is very dry, you may have to break the stool in half and take a sample from the inside of the stool. You can also wipe away any stool off the paper once it's been on for a few seconds, and then take a look at the color. You are looking optimally for 6.0 to 6.3. If you have a higher pH than this, then you might consider using our Phylamet microenema. Don't use an enema bag or a squeeze bottle. Take my word for this. Glass syringes are much better, and this is an example of one that we have on our site. They're much more comfortable than using the ones that you can find that are made of plastic. Consider doing the Phylamet microenema daily, checking your stool pH until you can get to 6.0 to 6.3. Then you can stop taking the microenema, and as long as the occasional tests with pH paper show that your microbiome has stabilized at 6.0 to 6.3, you're good to go. Some people might find that they need to do the microenemas longer or once in a while just as a booster to their microbiome. Another thing you can do is you can also do our Allergica product. Allergica contains allagitannins and gallotannins, and those two ingredients are known to both feed the good gram-positive as well as kill or cause to go into a suppressed state the gram-negative. There's a few other things you can do. First off, don't go low carb. Low carb can trigger good bacteria to go bad. Even bifidus can make toxic formic acid if the carbohydrate levels get too low. Instead of going low carb, consider going low glycemic index. 2, minimize gluten. That would be wheat, rye, and barley, because it causes the cells of the intestines and other membranes to leak. You don't have to be off gluten forever, but consider it for a week or two while you're getting your microbiome reset. 3, don't eat more fat than you can absorb. Remember, the inflammatory lipids that the gram-negative bacteria are made from are made out of fat and sugar. That's the LPS, lipopolysaccharide. The lipo is fat. 4, don't eat more protein than you can absorb, because any protein that you don't absorb in the small intestine spills over into the large intestine, and it can undergo putrefaction by gram-negative bacteria into the toxic amines. 5, try to limit your exposure to drinking and bathing in chlorinated water, but mostly drinking. Chlorine is one of the things that specifically kills gram-positive bacteria. 6, minimize alcohol. Alcohol also makes the stool go more alkaline. So now you know the secret of recovering your microbiome. I know it was a very complicated video, and perhaps you want to go back and listen to it again, but it really comes down to some very simple concepts. We need to get rid of the gram-negative toxic bacteria, and the way to do that is short-chain fatty acids in the presence of an acidic environment. So a microenema with short-chain fatty acids in an acidic concentration gives you both of those two things you need. It gives you the acids, the acidic environment, and the actual acids needed to deal with the gram-negative bacteria. And once the gram-negative bacteria are knocked down, the toxic gases back off, the toxic amines back off, the lipopolysaccharide inflammatory elements back off. All of these things start to get better for you. The other thing you want to think about is supporting the lactic acid bacteria in your proximal colon. So while I've been talking about recovering your microbiome, I'd be remiss if I didn't mention all the other amazing things that good short-chain fatty acids can do for us. Good short-chain fatty acids control the speed at which DNA is accessed, which is called gene transcription. Since most people are low on short-chain fatty acids, the good forms, this results in a slowing of their repair mechanisms by up to 50% or more. If your body needs to repair something, it has to get the instructions on how to do the repair. It's also for growth. If you have a child who's not growing properly, they may not be accessing their DNA in a timely manner at a proper rate, and so they might be growing more slowly, and that can be a lack of short-chain fatty acids. So by increasing your short-chain fatty acids, it's not that you become superhuman in terms of being able to repair. You become what you should be. You get your birthright, which is the ability to grow into a strong and healthy person, which is the ability to quickly and effectively repair injuries to your body. That's all about gene transcription. Good short-chain fatty acids also improve your mood and your mental function. They stimulate the vagus nerve, which puts us in a rest and digest state, and they also help maintain a proper blood-brain barrier, which keeps toxins from getting into the brain. So with good short-chain fatty acids from a microenema initially and from your own natural production over time, you may find yourself in a calmer state and with a brain that functions at a higher degree of capacity. You may find yourself with a greater ability to remember things, better focus, better mood. I've also noticed I feel much more satisfied after eating food. This is due to several reasons. Short-chain fatty acids are ketogenic and balance blood sugar, but they're also responsible for about 30% of our energy supply. What that means is if you don't have good short-chain fatty acids coming out of your microbiome, you've lost about a third of your energy. And then with the absence of the good short-chain fatty acids, you would have the bad ones. And so that's going to knock out energy because you have to deal with them. So if we lose a third by not having the good short-chain fatty acids, and we lose, let's say, another third by having to deal with the toxins of the bad short-chain fatty acids, well, that's 2/3 of your energy out the window right there because the gut's not working properly. It's really a miracle that we have inside of our gut. The microbiome is amazing at giving us metabolic balance and health and longevity and happiness. If you want to find out more about how our Phylamet short-chain fatty acid microenema may support your microbiome and improve your health, you can reach us by email or by phone. Take care.